already looked at three cases. The first case is when there is a dominant allele, there is a recessive allele, and when the offspring is formed, the dominant allele expresses itself, and according to the law of dominance, we get the doll phenotype, that is the dominant phenotype. Case 2 we have looked at incomplete dominance. What happens in incomplete dominance is that both the gene products are formed, but an intermediate like a red flower and a white flower is going to produce a blended phenotype, that is a pink colored flower. The third case that we're going to look today is when there is one allele and the second allele. In the offspring, both the alleles are present, but they are not going to get intermixed. They are also not going to show dominance and recessiveness. In fact, both will form their gene products and the phenotype will be a mixture of both. Hello guys, welcome back to Learning with Isha. The topic for today's discussion is case 3, that is codominance. Well, codominance is a phenomena where in a heterozygous condition, see the condition should be heterozygous, दो तरह के different alleles होने चाहिए. In such a scenario, expression of both the alleles is going to be visible. So a classic example of codominance is the ABO blood grouping system in humans. Let's take a look. The ABO blood grouping system was given by a scientist Carl Landsteiner in the 1900s. Let's try to understand the basis of this blood grouping system and then we'll look at how codominance plays a role here. So to understand it, let's draw a pair of homologous chromosomes. So one copy comes from the mother, one from the father in a diploid organism. So here on, let's say this is chromosome number 9 for humans. So this is chromosome number 9. So we have a gene located over here known as the I gene. So let's just draw the I gene. There are going to be 2 copies of this I gene. Now for this I gene, these 2 copies of the same gene are known as alleles. So they represent 2 alleles. Now the first question is this I gene, what does I refer to? The word I refers to isoagglutinogen. Isoagglutinogen. Okay, isoagglutinogen is another word used for the word antigen. Okay, but now what is antigen? So basically it's a molecule that is going to activate the immune system. So this I gene produces a sugar, a basically glycoprotein. So this produces a glycoprotein that serves as an antigen. Where is this glycoprotein located? It is located on the surface of the RBC. So this is my red blood cell and on its surface there is going to be a sugar. Now there are 3 different alleles of the I gene. So IA, IB and IO. Alleles represent alternate forms of the same gene. So basically they're all going to produce these antigens, but there's going to be a slight variation in the structure of these antigens. So in the case of IA, the surface of RBCs is going to have the A-type sugar, B may B-type sugar, and case of O, it represents a null situation. So I will have an RBC which is not going to have any antigen on its surface. So there's just going to be my RBC devoid of any such antigen. So this is what is basically happening on the gene level. So now where does codominance come into play? With this information, we'll be able to figure out that when I say IA and IB alleles are codominant, it means that the product of both these genes, as in the A and B antigen, both are going to be present on the surface of the RBC. So there's an RBC having the A type antigen and B type antigen. But in the case of the O allele, both A and B type alleles are completely dominant over the O allele. So let's say I have A allele or the B allele and I have the O1, it is completely going to be dominant over the O allele. So why is that? Basically, I'm going to have an RBC जिसमें O वाला तो कोई भी antigen contribute नहीं करेगा. Let's say I have the I type allele. So the A antigen is going to be present on the surface. That is why A and B alleles are codominant, whereas they are both dominant on the O type allele. Now with this knowledge, it would seem logical to write the different combinations of the alleles possible. Now we see that in a homologous chromosomes we have two alleles. इनका क्या combination है? We are going to try to write it. Now let's say from the mother I get IA. From the father also I get IA. From the mother I get IA, but from the father I'm getting IB. From the mother I'm getting IA, here I'm getting IO. Similarly, I'm going to go ahead and fill in this table. Now, in each of these cases, let's try to deduce the blood type that we are getting. So, the blood group in this case is going to be A. Codominance hoga, so AB. Here, completely dominant A over O, so A blood group. In this case, codominance, so AB. Here we'll get B. B dominant over O, so B. And O and O means O. So these are the different blood groups that are possible. So now a question that seems logical is here is how many different types of genotypes do I see? Well, you may think कि यहाँ पे तो मुझे 7 different types दिख रहे. In fact, you would realize that this case and this case is just the same genotype that has been reversed. So we will say that total we have 6 different genotypes that are giving me how many phenotypes? Well, they are giving me 4 different phenotypes. The phenotype in this case is the blood group and I get 4 blood groups. A, B, AB and O. So codominance का role था that genes IA and IB are codominant with each other. This ABO blood grouping system also serves as an important example of another genetics phenomena. It is known as multiple allelism. Well, what is multiple allelism is the topic for another video. A brief introduction of this would be that there are different alleles, like the I gene has 3 different alleles that are going to give me different phenomena. So in the next video, we're going to move on talk about multiple allelism. Please consider subscribing to the channel if you like this video. Do check out the other videos from the genetics series and write to me if you have any queries or doubts related to these topics.